Hello, everybody, and welcome to Scripps Research Fully Interactive Resources for Students and Teachers, or FIRST. My name is Rosa Alvarez-Seclar, and I will be your moderator this afternoon. If you're joining us for the first time, Scripps Research is one of the largest nonprofit private biomedical research institutes in the nation, and our mission is to develop the next generation of medicines and diagnostics to address the most present health concerns in the world, such as COVID-19. I encourage everybody to visit our website after the webinar, and if you have any questions, please don't hesitate to contact us. Before we move into the presentation for today, I'd like to remind everybody of a few housekeeping items. The format of the presentation is going to be a 10 to 15-minute presentation by our speaker today, followed by another 10 to 15 Q&A session, where we're going to be taking your questions live. For these, we ask you that you use the Q&A button on the bottom of your screen. You're going to see a menu, and you can type in your questions at any time during this presentation. Everybody's videos have been disabled, and you have been muted. So the only way that you have to interact with us is through this Q&A feature. If you are on Facebook Live, we ask that you please use the comments box to send us your questions or your comments. We also want to interact with you via polls, and I think that right now it's a good chance to practice that. So if you can please take a moment to respond to the questions that are now on your screen. These are, the first set of questions are to get to know our audience, and this is going to be important for our speaker today to be able to respond the answers appropriately, the questions appropriately. So I see that most of you are high school students, a few middle school students, and college students as well. And yes, you have heard from Chris, is divided, right? Almost 50-50 of yes and no's, if whether you have heard from CRISPR technique or not. And you're looking to learn about CRISPR today and advice on how to pursue science. Thank you so much for that. And without further delay, I want to introduce the speaker for today, Taylor Lloyd. She's a graduate student in the lab of Dr. Michael Boddy in the Department of Molecular Medicine. And as the title suggests, she will be talking today about CRISPR-Cas9, which is a genetic tool that has been making the news on the last several years, and it's a very hot topic. She will also be talking about her research, and you can ask her about her career so far and questions about going into grad school and into a research career. Thank you so much, Taylor.
Awesome. Okay, sure, I'm unmuted. Okay, so hello, everyone. As she said, my name is Taylor, and I'm a graduate student at Scripps. So today we're just gonna do a really quick 15-minute overview about CRISPR. And CRISPR, if you haven't heard of it before, is a genome editing tool. And so it can basically go in and make direct changes to your DNA sequence. So let's just start with like a really quick introduction on what is genome and what is DNA, what does it do, what does it look like, that sort of stuff. So first of all, you have a human, right? That's like us, that's like macro view. We all are familiar with that, I hope. But if you zoom in, like super, super close, you'll see that you are made up of cells. All living things are made up of cells. And these are basically just little individual packets that all operate individually, but come together as a whole to make life possible, to make it possible for there to be a human being, for there to be a dog, a cat, a plant, everything. So with a cell, so if you like zoom in on the cell, it has a bunch of, so this is like a view of 1, 2, 3, 4, it's like 7 cells, right? So if you zoom in on any of them, you'll see that inside the cell, they're like another tiny body. So they have organs and things on the inside of the cells. So like you are a human and you have a stomach and intestines and your brain and everything like that. The cell also has very similar organs, right? Not so much like stomach and brain, but like things that could be analogous if you wanted to extrapolate. And the one organ that we want to focus on today, I would say, is probably most like the cell's brain. And I've drawn it as a big blue circle here. So if you zoom in on the blue circle, which is the nucleus of the cell, you'll see that there are a bunch of like squiggly line things, basically, if you just look at this under a super awesome microscope. And each one of these squiggly lines we refer to as a chromosome. And so the chromosomes, maybe you guys have heard of chromosomes before, but they come in a set of 23, and well, it's different for every animal, but for humans, we have 23. And every chromosome has an identical buddy pair. So it has a complete copy of itself in every cell. So you have not just 23 chromosomes, but you actually have 46. Okay, so the second buddy of it, its identical copy is basically your backup copy. And that'll be important later when we talk about some other things, but I just want you to keep in mind that you have 2 copies of DNA. It's like having 2 copies of a single book, right? So if you zoom in on the chromosome, maybe I gave it away already, but if you zoom in on the chromosome, you'll find that this like blob, like squiggly bunched up line thing, that is all DNA. So chromosomes are just compacted DNA. And DNA is the subject of everything we want to talk about today, and it's basically just a string of molecules. And so it's made up, you've probably seen this nice like twisty situation before, and so the blue is called the ribose backbone. So it's basically just like a sugar backbone, not that important at this point, but it's the backbone, 2 of them, and then the 2 strands are held together by binding of molecules on the inside. So there are 4 different types of molecules that can link the 2 DNA together like a ladder. And we call these the base pairs. So if we just zoom in on this for a little bit, we gave every base pair, as scientists, we just gave it each a letter, because that's honestly the easiest method for keeping track of all of this. And so we basically have A, G, C, and T. And so your DNA basically is just a really, really, really long string of AGCT. AGCT, GGC, ACCC, and it's unique to everything and everybody. Everyone has very unique DNA sequence. And there are like bazillions and bazillions of these base pairs on a line. And every one of the 23 or 46 chromosomes is all made up of DNA base pairs, all the way down to the end. So like I said, we break this down even further. So this is just the base, one single one. And then when you have it bound to its partner, so G always binds with C, and A always binds with T. So if you have it bound to its partner, that's called a base pair. And if you have 3 in a row, we call that a codon. And so if the base pair is like a single word out of the dictionary, a codon is like a sentence. And so it's like a combination of a lot of words to have it make sense. So the cell can read a codon and it makes sense. It tells it to do something, right? And if you have a ton of codons all in a row, we call that a gene. And so that, I don't know if you guys can see that. Okay, great. So that, if the codon is a sentence, so if you have words that are base pairs, sentences that are codons, a gene is like an entire instruction manual book. And this tells the cell how to perform certain functions. So if something's happening to a cell, like say you got infected with a virus or something, the cell, in order to respond, will go back to the DNA and say, what the heck, what do I do? And then it reads off the DNA, the genes that it needs, and the genes tell it what to do and how to respond to that problem. Okay, hopefully that part makes sense. That's essentially what is DNA and what does it do. So that leads us to what we refer to as the central dogma of molecular biology. And so in molecular biology, there's basically 3 types of molecules that we are really interested in. So the first one is DNA, which I just explained. It's like 2 backbones bound together by base pairs. And then there's RNA, which is very similar to DNA, but it's usually just single-stranded. So it has one side of the backbone and then one single side of the base pairs all the way down. And then we have proteins. And these are made up of what are called amino acids. And this is basically the machinery of the cell. This is what carries out all of the functions in the cell. So DNA is like the archive, and then it makes RNA, which is like a photocopied page, which tells the cell how to make the protein. So this basically, the central dogma is that DNA encodes for the RNA. So if the DNA is like a really great instruction manual, the RNA is like a photocopied page out of that instruction manual, so that you can take it wherever you want, you can use it, and then you can just get rid of it because you don't need it anymore. It's more temporary type of information storage. And then from the RNA, that page of the instruction manual can be used to synthesize proteins. Okay, so this part is called transcription, and this part is called translation. So it's basically like photocopying and then using that photocopy. There's a couple other steps to this. So this is the central dogma. This is like all that you need to remember about molecular biology, basically. But there's a couple other extra pieces. Number one, DNA can remake itself. So you can actually have DNA encode for more DNA, which makes sense, right? If you want to reproduce cells, like if a cell divides, if a cell divides, if you have a baby, if anything, you need to make another form of life. You need to have more DNA, right, to make more cells so that life can continue on. There's also some special cases in which RNA can then encode for DNA. And that's not usually what you want to happen. A lot of time viruses come as RNA, and then they can be what's called reverse transcribed into DNA. And then you are infected, and that's not so great, right? Okay. So, central dogma. Basically, the point that I want to make here is that DNA is the top of this chart. It's like the apex predator of this whole thing. If you change something that happens in the DNA, you are going to change what happens in the RNA, and that will change what happens with the protein, and that means that the cellular response is going to be different. So you have eye color is like really nice and easy genetics, right? So if you have blue eyes, that's encoded in your DNA, read off by the RNA, and translated into blue eyes proteins. But if you had a change here, something happened to your DNA, and it had a mutation, you could then end up with a brown eye mutation, which would then be transcribed into brown eyes RNA, which would create brown eye proteins. And then thus you would be brown eyes. So the DNA is much more than just your eye color, of course. It's literally everything about you. It's how you stay healthy, how your body functions. So it's really, really, really important to keep DNA safe and stable and exactly as it should be. But the bad news is that your DNA can be damaged, and it is actually damaged a lot, which is not great, right? So there's a few different ways that this can happen. The first is that you can have damage to the actual base pairs that are in between the two backbones. And so this is basically inside the coding region. You can have the base pairs be mismatched. So I said that, G always binds with C and A is always with T. So you can have a problem where like they have the wrong partner, G is to T or A, or vice versa. And that is not good, and it represents a problem that means that coding information is being lost. You can also have base pairs stick together. You can have other molecules, like maybe you ate something toxic, get stuck inside your DNA, and that can all cause there to be loss of information. You can also have damage to the backbone of the DNA. So, of course, you can have a single-stranded break, so just one side of the backbone has a gap in it, some sort of break, something happened to create this problem. And this is not so bad. You can usually just come in and glue these two together. That's not so bad. There's no information that's being lost here. Everything can be repaired. You still don't want this. There's always problems that can happen after that, but this is not as catastrophic as a double-stranded break. So if both sides of your backbone are broken off, this creates a really, really serious problem. And so let's just think of if you have a bunch of DNA broken. So this is all just fragmented. Think of it like a sentence that is getting fragmented. So Nithya, can you run the polls that I gave you, the first poll? So basically what I want to ask you is that this is a sentence that has been fragmented. So how would you repair this fragmented sentence? I'll give you guys a few minutes. Hopefully this is not too hard. Okay. Awesome. So most of you guys got it. This says biology is great, right? Biology is great, right? Okay. Nithya, can you run the second poll? Okay, so imagine instead that you just have a series of base pairs that are broken. How would you then repair that? You don't have the luxury of having the English language guide you anymore. You just have chemistry and molecules. And they actually don't care which base is bound to which. Okay. Awesome. So this one, we have a slightly more divided response, I can see. Most of you guys are, I mean, it's a little bit all over the board, but right, you can't really tell. Once all the bases are sort of dissembled like that, it's really hard to tell. You can only hope that they are already situated in the way that they were before they broke. So a lot of you guys just stuck that sequence together like as a big string. That is what the cell does, and it hopes that it's in that way. But it could be that those guys got scrambled. It really could be any situation here, which makes this really catastrophic. If this is an important gene, if this is the gene that lets your lungs function, and it gets broken like this and scrambled, and then the cell is really not sure how to put it back together, that is going to be a major problem. That could really impact your life and your ability to go on, right? And DNA is can be broken, I should mention this, in a variety of ways. And so maybe some of them are like kind of alarming, and also alarming that this is happening to you all the time. So sun exposure, sunburns, is actually fragmenting your DNA. So when you get burned by the sun, that burn response is your cells trying to repair and deal with the UV damage that you dealt to the DNA. Usually that results in single-stranded breaks, but it can be double-stranded as well. If you stood out in space and just got a bunch of space irradiation on you, you would totally fragment up your DNA in double-stranded breaks. When people get chemotherapy and get irradiated, I don't know if you guys have heard of that, but if you get irradiated on purpose for medical reasons, it's also completely fragmenting DNA, which makes it very dangerous as well. And then there's certain things that you can eat and chemicals that you can come in contact with that can also cause all these problems, right? So it's good to wear sunscreen and keep yourself safe, right? But this is really happening all the time. You can't always stay out of the sun. You can't always have an umbrella or whatever. So luckily, the cell does have ways to deal with DNA damage, which is great. Otherwise, life really would not survive. So there's two main pathways for DNA repair. And the first one that I want to talk to you about is on the left, so just focus on this side for now. It's called non-homologous end joining. And that is basically non-homologous means not matching end joining. So end joining is end joining. And basically if you have a break in the DNA, so something happens, maybe you got a sunburn, who knows, the cell will basically look at the ends here. And DNA doesn't always break off nice and flat and straight. So the first thing that it's going to do is polish up the ends and make them really nice and flat and straight. And so that might mean cutting some stuff off or adding some stuff in. And then it's going to put glue basically on the ends of both and glue them together, or fancy word for that is ligation. So you get end polishing to make them nice and flat, and then you get ligation. And this will just glue it right back together. But because of the end polishing process, you can have mutations added. So I said that sometimes they cut things off, the cell will cut stuff off, and sometimes it adds it in order to make them nice and flat so they can be glued together. That is referred to as error prone. So that's like certainly going to introduce problems. So if we have our sentence from before, biology is great, and then there is a break, maybe like here, the cell will look at this and then it's like, all right, I need to polish this up. So maybe it gets rid of this O and this L, it doesn't like that either. And then it's like, I don't know, maybe I took off too much, so I'm gonna add a B and an R. I don't know. I don't know what's going on. It's a break. I'm just trying to fix this. So then you are left with bio free is great. And that is not great, right? So that is an information loss, and it's actually also an information gain of wrong information. So it's not only did the cell forget something, it also learned something wrong. So that is not great, that is error prone, and so you definitely want to avoid getting this happening in something that is really vital. Luckily, a lot of your DNA doesn't directly code for vital genes. So this is mostly an okay process, and usually everything ends up being fine. But there is a more error-free method, and so at this point, focus your attention on the right-hand side. So this is called homologous recombination. And so homologous just means the same. So where this one was non-similar end joining, this one will be similar recombination. And that just means like rejoining and copying and things. So what happens basically here is that you get a cut right here. And then this side will, so I told you, if you remember, that your DNA has an identical copy. It has a bud that should be exactly the same as before. So in this case, the cell says, all right, I know that there's an identical copy around here somewhere that I can borrow, and that has the information that might be missing, and it'll make sure that everything makes sense and is correct here. So what this side does is that it will go down to its buddy and start copying from here. So it uses its identical template, identical copy as a template to fix this break. And what that ends up with is that you have your identical copy, which is fine, nothing happened to it. And then you have the broken copy, which will now incorporate a little bit of this identical template copy into it, right? So it just copied and pasted it up here. And this is great. This is like totally going to be way more error-free. So things will be much more high fidelity in this case. But there is still problems associated with this. You don't want to run outside and immediately get sunburned or eat a ton of chemicals, because if there is a problem in this template, say you have a mutation here that wasn't affecting you because you had a good copy here, right? So let's say you had a disease mutant that this is non-functional, but this one is functional, so that's fine. What happens in this case is that this non-functional copy gets copied over, and now you have no functional copies of this gene, and therefore you will get whatever this disease is. So that is still not the greatest option. The best option is always not to have damage. But if we think of it as a sentence, biology is great. And then we have a cut that's happening here, same spot. And if you have the identical template down here, but it has a mutation in it, something that just doesn't really make sense. It's not a good sentence, but it's there. That will get copied over, right? So this side will say, oh, okay, so there's a cut here. I don't know if I lost information. Let me go start copying from here. And then I'll come back to my own. And that will end up giving you two copies that say, fire tea is great. Sorry, my handwriting is getting like really messy from this, but. Right, okay. So this is called loss of heterozygosity. And this is one of the ways in which cancer can be driven as well. Okay, so before I move on, I will answer this question that we have. So why you get freckles after sunburn? Okay, so I'm not a dermatologist, but I believe the reason you get freckles after you sunburn is sort of due to DNA damage, yes. So it's when your cells get damaged, the DNA becomes damaged, and then part of the response to that is the production of melanin. And so I did learn this in undergrad, but I forget now. But basically, I believe that melanin is produced, and then it just happens to hang out there. And I think that that's meant to be more of a protective measure. So if you have melanin, it will protect your cells a little bit better from sunburn. So it's not like it is a direct because of you got DNA damage, but it is like a side effect of the cellular response rather than being directly involved in DNA repair. Hope that makes sense. Okay. So you can ask another question if that totally didn't make any sense. But from here, I just wanted to get you guys familiar with DNA repair from this. And what I want you to notice here is that in both cases, mutations and changes are able to be made directly to the DNA sequence. And so this is unintentional genome editing, right? You're changing directly the DNA itself and the coding in the DNA, but this is completely unintentional. You just damaged yourself and the cell is trying to fix itself. So how then do we start to intentionally edit your genome, maybe using CRISPR-Cas9? So enter CRISPR-Cas9. So CRISPR-Cas9 is actually a bacterial protein, and CRISPR stands for clustered repeat interspaced, or clustered something, I don't know, it's like a bacterial protein. I should definitely know what it is, but it's not important in mammalian cells what it stands for exactly, because it doesn't do the same thing in mammalian cells. Or it does, but it's not for the same purpose. So bacteria use this protein as a tiny immune system. And in studies with mammalian cells, like what I work on, we use it as a genome editor. So that's like a slightly different purpose. And it's made up of the Cas9 protein. So I just do that in green. All the proteins are green here. So Cas9 protein. And then it has something called a guide RNA. And it's different in bacterial cells. It's like a slightly different system. So this is actually synthetic for the way that we use it in mammalian cells to make it like as easy as possible for us to use. There's lots of technical conditions for basically running experiments that has led it to looking like this. So you have the Cas9 protein and you have what's known as the guide RNA. And so the guide RNA has this like double stranded structure, so it like binds to itself here, and it makes this weird little like 3D folding thingy so that it fits and binds into the Cas9 protein, so that these guys basically come together like two puzzle pieces. It's great. And then it has this other section over here that is really the guide part. And so there's a big string of base pairs that are open, and this will be used to guide it directly to the sequence of the DNA that you want to edit. So I'm just gonna write in the sequence. So from the guide RNA, I know exactly where this is going. And it'll intercalate into here. Okay. Yep, base pair is here. Okay, so if this is your genomic DNA, the guide RNA is complementary, so it specifically binds to one particular sequence in your genomic DNA. So what this does is brings the whole Cas9 protein, which it's stuck to this sequence. That's basically the entire way that works. And what does that do? Okay, so now that's what the guide does. Cas9 has just been along for the ride so far. So what does Cas9 do? Cas9 induces a break. It cuts. It's like a nuclear scissors. It just makes a break in your double-stranded DNA. And then you have specific cutting, which can then be directed towards either pathway of the DNA repair machinery, which I just talked about. So in theory, if you have a gene that you are interested in, especially if you're interested in studying at this point. It's mostly for study than for treatment, but it could be for treatment. So if you're interested in gene X or whatever, you can use the guide RNA to direct the Cas9 protein to gene X. It will induce a cut, and in this cut, which is now in the gene of interest, will then be repaired. And it can be repaired either by non-homologous end joining or by homologous recombination. So if the cell basically gets to decide. At this point, there's not a lot that we can do to direct it. There is like some compounds and things and other strategies that people have gotten fancy with, but for the most part, the cell basically has a choice here. And if it decides to go through non-homologous end joining, if you remember from back here, non-homologous end joining basically looks like end polishing and then gluing it together. It will start to insert little changes. And so this little insert is uncontrolled. We don't know what the cell is going to add here, but it will probably add something. And if you only care about getting rid of this gene or making it non-functional or messing it up in some way, but you don't really care how it's messed up, you just want to like mess with it, this is a great option. This will just start to induce like little random changes, and usually just like 1 or 2 bases being off will totally throw off the whole system. So if you wanted to make a knockout, say you had a black mouse and you wanted to make it a white mouse, you can just cut the black gene. I mean, it's a little bit more complicated, but you cut the black gene and then let it repair through non-homologous end joining. It'll insert some little things inside of the black gene and it'll make it totally non-functional, and then you end up with a white mouse. Okay, but that's not always that useful, right? So say you had a white mouse and you wanted to make it a black mouse. That would not be possible here because this is totally uncontrolled. It just is going to add some stuff. So how do we start to direct the editing that we want to happen? And that is through the use and sort of exploitation of the homologous recombination pathway. So directed genome editing. Okay, so if we make the cut, you get broken DNA. So I'm just going to draw just 2 lines here. And then it can go through homologous recombination that uses an endogenous template. So this endogenous template, I just mean like it's from the cell itself. So this is the copy that all of your chromosomes have an identical copy. So this is just what I showed you before. It uses the identical copy, it reads off of the inside, and then it repairs itself just the way that it was before, no errors induced. Okay, so that's not that helpful. If you wanted to make a change that's specific, and then the cell just fixes this as like perfectly, that's not exactly what you wanted, but that is what biology wants. But you can sort of manipulate this process by adding an exogenous template. So if you have sorry, this is like super messy, but exogenous template. So this is something that we would just give the cells. So at the same time we give the cells Cas9 in the guide, we can give them an exogenous template that is homologous to this region around the cut, but includes whatever change we want to make in the center. Okay, so then now the cell has 2 choices. Either it can use its endogenous template, which we can't get rid of, that's just going to always be there, or it can use our new exogenous template that we give it, or synthetic template, really. And then that will allow it to be repaired with the change that we want to make at the center. And so now, if you wanted to make, if we go back to our mouse, we could make a white mouse black. If we guide this to the black fur gene, we make a cut. It could always repair it using its copy of the black fur gene, and then you get no edit. Nothing happens, that's not that exciting. Or we can give it an exogenous template, which has the change here that gives it a white, or a black fur gene. So if this is a white mouse and you want to make it black, background. So black, the white gene you cut, you give it an exogenous template that includes the black mouse gene, the black fur gene, and it will incorporate black fur gene into the section of the white mouse gene. Therefore, you get a black mouse out of a white mouse. And that is basically how we are using CRISPR-Cas9 in the lab right now. And so I do a lot of study on cancer cells and things, and so I want to know what cancer cells require, what they don't require, and I will purposely change or mess up the genome to investigate that. So say there's gene A, and I think that that is required for cancer cell survival. I can go in and specifically edit it. I can knock it out completely. I can alter it just slightly. If I think that one part of the gene is more important than the others, I can just alter that. And it's basically been a really invaluable tool for being able to directly probe genomes and exactly what parts of the DNA is important. And then because you're changing the DNA, you can change everything downstream as well. So if you're interested in the RNA and what parts of the RNA are important, you can make changes using Cas9 to the DNA that will be reflected in the RNA, and similarly with the proteins. So the black fur, white fur, that's actually at the protein level, right? So that just means that the DNA is creating a different protein out of that. But the hope is that eventually this will be something that we can use in clinic to help humans. So if you had a genetic disease that, I mean, there's lots of genetic diseases, right? You guys probably know of like a few, like my family has hemophilia that runs in it, right? So I don't have that luckily, but that is a loss of function. The hope is that hopefully you could go in and directly edit that gene that is messed up to add a functional copy of that gene. And then ta-da, you have a person who's completely cured of their genetic disease, which was, to this point, not something that was fixable, only treating the symptoms, right, not the cause. So a lot of times the DNA and the gene is the cause of the disease, and you can go in, hopefully sometime in the future, and fix it using CRISPR-Cas9 to edit the genome. They're doing a few of these things, so there's not a lot because people are concerned about off-targets. The efficiency isn't great yet, but there is, I think, a clinical trial working on sickle cell anemia, which is also similar in that it's a loss of function as well, and I think they're trying to insert a functional copy or rearrange it to use something functional instead. Yes, and so that's where I kind of want to end. I hope that wasn't like too long, and I can answer your guys' questions.
That was great. Thank you. Yeah, it was awesome. I think that everybody enjoyed that. I think I'm mindful of the time, so let's go ahead and get some of these questions in. And I want to let everybody know that if we don't get to go through all the questions, then we'll add them to the website, and Taylor and other students will answer them. There's another poll on your screen. If you can quickly take this one as well. This is for us to know how we're doing so far and what else you would like to hear about. But meanwhile, I'm gonna start reading the first question. If someone who has two different eye colors, does that mean they could have a mutation in their DNA?
Yeah, so for sure. So eye color is way more complicated than being a single gene. Geneticists and biologists like to be like, oh, you have a blue color gene, you have a brown color gene, whatever. But there's actually like, it's a whole network of genes that have to work together to give you your eye color. I'm not informed enough to tell you exactly how you end up with two different eye colors, although that's like really interesting. But yeah, that's definitely going to be stemming from a mutation. I would be really surprised if it wasn't a mutation in the DNA. We'll say that.
Great. The next question, I heard of the one mouse that had something like a firefly gene and it will glow green. Was that using CRISPR-Cas9?
Yeah, so I'm pretty sure that that was engineered using CRISPR-Cas9. There are other ways to make changes in the genome, especially with mice that have been used for a really long time. But those are super duper inefficient. And so it could have been made using one of those old school ways, but it was definitely a genetic manipulation that was making it glow green, which is really cool. They made rabbits too that are glow in the dark.
Great. How do you know if the cell is going to use homologous or non-homologous repair?
Yeah, so that is like an awesome question, and it totally depends on the cell type. It depends on the type of DNA that you are trying to edit. So there's some DNA that's more like condensed and some that's more open. And so the open DNA usually is fixed using homologous recombination, and closed is usually through non-homologous end joining. And so that just refers to like like if you had a slinky, is it like stretched out or like collapsed together? Basically, it's almost exactly the same. And then also cell cycles. So when the cell is dividing, it goes through a few different phases basically to ensure that it's replicating itself correctly. And it is more prone to using one or the other in each of those phases. And so you can kind of like, there's ways to like direct it towards one or the other by saying, hey, why don't you hang out in this one cell cycle for a little while? Or I'm going to make my life easier by using this one cell type that likes to use homologous recombination a lot. But that is something that is struggled with a lot in science.
Thank you. The next question, could this technology be abused?
Yeah, so for sure this technology could be abused, right? It's all the editing in that is like legal and ethical at this point has been on mice and human cells that are in culture, but there's no reason that we know of that you couldn't edit like a human embryo, right? So then you could have, that would be awesome, especially for parents who have genetic diseases or they want to be parents, but they have a genetic disease and that they don't want to pass on to their children. And so that would be great because then you can go in and you can fix that and you can ensure that the child will not have that disease. But it can be abused in that you have parents who are like, well, I don't want to have a blue-eyed baby. I want to have a green-eyed baby. And so then you can go in and fix that too. And that's not really what scientists are hoping that it will be used for. It's not approved on humans like at all. There was a scientist in China that totally off the grid edited a human genome and the children are living and it's intended to fix a genetic problem, not so it would be like one of those, we hope that this would be an ethical use, but it wasn't done through the correct channels and so it still is unethical. And as far as we know, the babies are okay, but like you don't really want your children to be like monitored by the scientific community either. So I don't think they've been super free flowing with information. It's like hard to tell. But yeah, that's definitely a concern.
Thank you. So next question, when it comes to directed genome editing, has it become specific or targeted, or is it still a cat and mouse strategy?
Yeah, so this is, with directed genome editing, it is, so because it's directed, it's like quite specific. There are a lot of studies coming out now that are looking for off-target effects. Because of the guide RNA, it's really, you can direct it exactly to what you want, but what you don't know is whether you're directing it to other things on accident as well. And so that is something that people are really investigating heavily, is like the off-target effects at this point. So I'm not sure exactly what you mean by cat and mouse strategy. I hope that that solves it, but there are ways to sort of unbiasedly edit different parts of an entire genome to basically screen for genes that are important or unimportant and that you can use for CRISPR-Cas9. So if you wanted to say like what genes are important for living, you can knock out every single gene in the genome using CRISPR-Cas9 in a different cell. So you have like there's like 20,000 genes in a human genome that code for something. You have 20,000 cells and each one of them has a different knockout. And then you can see which one of those guys die. So that's like a non-specific, that would be more like a chasing after something strategy than a specific, like I'm interested in this particular gene.
Yeah. Thank you. Next question, can they now look to see if there was damage by X-ray? Can it be fixed now?
So I think X-rays that you get when you are, like if you're at the doctor or whatever, that is not enough usually to cause any like lasting damage. I think if you got, like if you're an X-ray technician and you X-rayed yourself every single day of your whole life, that would be a problem. But for most of the people who just get an X-ray or whatever, it's usually not enough damage that would be something that you could detect or that would be a problem later on. We have really great sequencing abilities now. And so you could, if you really, really wanted to, sequence your whole genome. And you could sequence like the part from where they X-rayed you on like your arm. You could sequence your arm cells versus like your leg cells where they didn't X-ray you and look for differences. Although, yeah, I don't think it's strong enough to really cause anything that's that big of a difference.
Thanks. Next question. How does the guide RNA find the exact location on a chromosome of the gene that is trying to cut?
Yeah, This is a good question. So there's, people aren't sure whether it like follows it, like if you had a string, if it just like follows it all the way down the string and until it like latches onto the part that it finds, or if it's more like diffuse, if it just sort of like falls onto different parts of DNA until it sticks to one of them. I think we're not, as far as I know, we're not sure exactly how that's working yet. But it is a good question. At this point, I would say it's probably, we usually overexpress. So we have like tons of copies of Cas9 and the guide and everything. So we're just like upping our chances that by random chance, if it's occurring by random chance, that it would find itself there or find its target. Yeah.
Great. The next question is, I'm going to try to go through some of these. I don't think we're going to be able to do all of these. So let's go at this one. Is CRISPR-Cas9 edits a gene. Will the gene be passed down?
Yes. Yeah. So I was actually talking to a taxi driver a while ago, or an Uber driver or whatever. And they were like mind blown that when you edit if you wanted to edit your children's genome, that would be in all of their children too. So any changes you make to the DNA will always be passed down. So going back to ethical issues with DNA editing, any changes that you make will be preserved in the human gene pool forever, unless those guys that have that change like die out for some reason. So if you make a change that ends up being really bad and makes people sick, but you don't know it for a while, that could be passed to a lot of people before you catch that, which is another thing that people want to really keep an eye on.
So that's great. Another question, what other scientific uses might CRISPR have beyond genome editing?
Yeah. So this is a great point. I was actually going to mention this too. So CRISPR basically works by directing a protein to a section of the DNA. That's like basically what it does. We have a bonus of genome editing because the protein Cas9 has cutting capability. So it can cut the genome there. But you could put whatever you want stuck to the Cas9 and take away its cutting ability. So we have something called dead Cas9. So it just goes there, but it doesn't do anything. It just goes. And so you can fuse that with like fluorescent proteins, and you can watch where the DNA goes over time in the cell, which is super cool. You can make basically like a lock and key on two different Cas9s. And so you can direct them to two different sections of the genome and then force them to like meet in the middle and come together, colocalize, which is also really cool. I mean, there's like so many really valuable research tools that can come out of this. Basically, anything you can think of where you're taking a protein to a section of the genome, it can be done with CRISPR-Cas9, I'm pretty sure.
Thank you. I think at this moment, I think we're going to wrap up. I want to thank Taylor so much for her time. I know it's her birthday. Happy birthday. She did a great job. It was awesome. And I can tell that this was a very popular webinar because there's still a lot of questions in the Q&A. Like we said, or I said before, we're going to answer all of them and they're going to be on our website. So at this point, I'd like to ask everybody to take the last poll and to let us know how we did. And also, I'd like to tell you that there's going to be another survey after we end the call, and we appreciate if you can also take that one. And there's going to be a space for you to type in any topic that you would like to hear in the future. So these today is the last webinar for the initial phase or series of the first webinars, but we're planning on having 2 more series during the summer. So please visit our website to learn about the schedule. And I'm sure that they're going to be interested in you. A lot of them are going to be on careers in science and also other workshops on how to apply to internships and to grad school. And so I think that everybody will enjoy those webinars as well. And with that, I'd like to thank Taylor again and everybody for joining today. And I'll see you next time. Thank you.
Thanks so much for your attention.